cross-bridge formation. So excitation, contraction, they have to be coupled together. The action potential depolarizes DHP, dihydropyridine, which opens the ryanodine channels and lets calcium come out. Calcium binds to troponin, the regulatory proteins then shift off the binding site and cross bridges are formed. When the regulatory proteins come back on, then relaxation phase occurs. When does relaxation occur? When the action potentials disappear. When do action potentials disappear? Back at the neuromuscular junction, acetylcholinesterase has broken down acetylcholine, and your local potentials are now below threshold. Cross bridges are formed when there is ATP present. ATP will bind to myosin's head and will get converted to ADP and PI. That provides the power, the energy for the power stroke. It can't be utilized until myosin binds to the actin. In order for actin and myosin to interact, calcium has to be present. For relaxation to occur, calcium has to not be present. So ATP again is required to get calcium back into the SR. Back at the neuromuscular junction, the local potentials get below threshold when the sodium-potassium ATPase starts to win. And that occurs when acetylcholine is no longer binding to the channels that are allowing sodium and potassium to move. Skeletal muscle has limited ability to store ATP. Slow twitch muscles can make a lot of ATP and keep it produced because they have a lot of mitochondria. So other muscle fibers, the 2A and 2B, have limited ability, either medium or limited ability to produce ATP because they're operating more glycolytically. So they have to have a source of glucose to run glycolysis. They have to store glycogen. So muscles that are glycolytic, they're going to have limited production of ATP and therefore limited tension development. When you have mitochondria and can produce ATP oxidatively, then you have a steadier supply of ATP. You can do more work for a longer period of time. All muscle fibers have some creatine phosphate that has even higher energy level than ATP. Creatine phosphate can donate one of its phosphates to ADP and regenerate ATP. That usually is only providing about 10 seconds worth of ATP regeneration. Then you either are out of creatine phosphate, and then you have to either stop that work or go to oxidative phosphorylation. So creatine phosphate releasing its energy by hydrolysis of the phosphate off of creatine phosphate and then transferring it to ADP to generate new ATP. So it's a very quick way of regenerating ATP as opposed to glycolysis, which takes longer, or oxidative phosphorylation, which takes even longer. But you will run out of that creatine phosphate very quickly, about 10 seconds of creatine phosphate energy replenishment of ATP during exercise. So mostly for very short bursts of energy. About 60 seconds between glycolysis and creatine phosphate, you can carry out maximum power output probably for about 60 seconds before appreciable de-recruitment occurs. Oxidative phosphorylation, much slower process. You have to go through glycolysis into the mitochondria, through the Krebs cycle, then the electron transport chain, and during all these this process you make high energy molecules which produce a lot of ATP, but it takes time to do that. So light to moderate exercise, you can do oxidative phosphorylation and maintain the power output. So endurance kind of activities, you use aerobic. Aerobic means with oxygen. So you're using your mitochondria to produce the ATP for those types of activities. Glycolysis will be the only source of power if the oxygen delivery to the muscles disappears, or if you have fewer mitochondria. So muscles that are ischemic, meaning they don't have the O2 being delivered to them at the rate they want to have O2 delivered so their mitochondria can produce the ATP, they will switch from oxidative phosphorylation to glycolytic production of ATP. So if you are doing high intensity exercise, you're more likely recruiting the type 2B fibers, which are glycolytic. The type 2A fibers that are oxidative and glycolytic, they will produce power, but if they become ischemic, they're switching totally to glycolytic ATP production. And if your slow twitch muscles are also getting ischemic, then the stabilization of your joints get all out of whack, and your application of power then is less efficient, and then you run into difficulties with the form and the maintenance of your body positions for maximum power application, and pretty much you're either going to get injured or your brain is going to say, hey, shut down because we're fatigued up here. Fatigued here, you shut down the activity of your muscles. So we've got several sources of ATP that production that we can utilize. We've got muscle glycogen. The type 2B fibers will store the most glycogen. All muscle that stores glycogen can only use glycogen for its own purposes. It will take the glycogen, break down the molecule to glucose, and then glucose can go through the glycolytic process. So you produce ATP that way, but you're pretty much netting 2 ATP at the end of glycolysis. So it takes ATP to break down glucose, and then you'll generate ATP, but you'll only net 2 per half of the glucose molecules. At the end of glycolysis, you produce pyruvate. Pyruvate building up will shut down glycolysis. This is end product inhibition. So pyruvate concentrations go up, then glycolysis ceases. If you have mitochondria, then pyruvate goes into the mitochondria, gets converted to acetyl-CoA, and you get more ATP produced that way. Another way to get rid of pyruvate is to change it into another molecule. You don't have a mitochondria in the cell, or you don't have enough mitochondria, and also in glycolysis, from glucose to pyruvate, you have to transfer a hydrogen to NAD, this intermediate step of glycolysis, you run out of NADs, your regulatory enzyme, then glycolysis stops. Pyruvate, your end product, builds up, glycolysis stops. So the body's figured out a very simple way to regenerate NAD and get rid of pyruvate. Take the hydrogen, stick it onto pyruvate, and now you have lactate. You have lactic acid. So take the pyruvate, make it into lactate, and problem solved. You got NAD back, so your high energy molecule is regenerated, your end product has disappeared, and glycolysis can continue. Lactate can be consumed by type 1 fibers, slow fibers, in your muscle fascicle. So it diffuses out of type 2 fibers, gets consumed by type 1 fiber, problem solved. So type 1 fiber takes it up, takes the hydrogen off, puts pyruvate into its mitochondria, makes ATP for itself. If lactic acid concentrations are so high that lactate makes it into the blood, it travels through the blood, heart takes up lactate, uses it for energy. No other place in the body uses up the lactate, it goes to the liver, and the liver will take lactate and make new glucose out of it and release it into the blood. If you run out of glucose, then your body is going to look for another alternative source for ATP production. Your type 2B fibers are pretty much out of luck when glucose limits are reached. But type 2A fibers and slow twitch fibers, they can go to fatty acids and go into lipolysis and beta oxidation of lipids and create ATP. And if you're not using your protein, but if you're exercising, you are, but if you're not using your protein, then your muscles will be catabolized for energy. So you must use your protein in order to spare it from energy production. So limited use of amino acids and protein in active skeletal muscle, and primarily go to beta oxidation, fatty acid synthesis, and that has to occur in mitochondria. That's why type 2B fibers are out of luck, because they have very few mitochondria. The ATP that's produced is then used for your cross-bridge formation. The relaxation is also requiring ATP, because you've got to get the calcium back into the SR. So for contraction and relaxation, you have to have ATP. Regeneration of ATP to maintain about 60 minutes of maximum power output can be assisted by the ATP of glycolysis and the creatine phosphate that stores energy in the form of that high energy molecule, transfers the phosphate to ADP, and regenerates the ATP very quickly. Need that enzyme creatine kinase. The highest creatine kinase concentration is going to be in a type 2 fiber. Highest in 2B, medium in 2A, least amount in type 1 fibers.
The first energy source to be tapped for muscle contraction is creatine phosphate. The enzyme creatine kinase transfers a high-energy phosphate from creatine phosphate to ADP. The products of this reaction are ATP and creatine. During prolonged moderate exercise, aerobic respiration fuels contraction. The first fuel to be tapped for this pathway is the glycogen stored in the muscle and the glucose delivered by the blood. If activity continues, fatty acids become the main fuel source. When exercise exceeds the capacity of the respiratory and circulatory system to deliver oxygen, ATP can be supplied by glycolysis alone. However, this is an inefficient pathway. Glycolysis produces only 2 ATP per molecule of glucose broken down. Aerobic respiration yields about 36.
Okay, so muscle cramping, muscle diaphragm spasms, things like that can be that you haven't, your type 2 fibers, which are your breathing muscles, have maybe a lack of blood supply due to this overexertion, and they've gone from aerobic ATP production to glycolysis, so they're less efficient in contracting, and maybe they'll cramp up on you. So muscle fatigue, there are a lot of theories out there, and none of them have been absolutely proven nor disproven. It's still a wide open area for research, so if you're interested in fatigue and you want to do research, go for it. So muscle fatigue during exercise is when the muscle no longer can contract, can respond to the stimulation. And this can be due to a central nervous system fatigue. Central nervous system fatigue is kind of a protective reaction to events that are going on in the muscles. If the muscle stability, if the joint stability is in question, if it's becoming wobbly, then the brain says, it's injury down, if you continue to do this, you're going to injure yourself, best to stop. If there is burning sensation out there, that lactic acid, then brain says, ew, doesn't feel good, let's stop. And this is protection, because if the lactic acid is causing this burn and you can't work through that burn, and you probably aren't trained enough to work through the burn. If you are trained enough, you ignore the burn and you keep on going, and as long as you're efficient in the muscle contraction, you won't fatigue. You continue to put out the same level of contractile force. When you are then if you push through the burn and you're not ready to ignore that burn, you're probably going to end up damaging connective tissue, ripping muscle too, but more seriously is damaging the connective tissue. Muscle cells reform by myoblasts forming new muscle cells pretty efficiently. As long as scar tissue doesn't get in the way, new muscle cells will be formed. But if you damage connective tissue, it's a very slow process to reform and get the connective tissue strength back. So central fatigue is kind of a protective response to the inefficient power output that's occurring out in the periphery. Post-exertional oxygen consumption, this EPOC. This is where the body continues to consume extra oxygen, even though you don't think you are consuming extra oxygen because your respiratory rate, your respiratory depth has gone back to normal. Your breathing rate is back to 15, your volume is back to 500. You cellularly, you're still consuming more oxygen after moderate to heavy exercise for the next day, sometimes 2 days. Because you have a lot of your exercise has taken you so far away from your homeostatic norms, and with all the waste product production, and you gotta clear that, and rebuilding your glycogen sources, and all of the things that are getting you back to normal homeostatic ranges, that continues beyond the exercise event. So they've actually measured this, where they've measured the cellular respiration and the oxygen consumption at the alveoli, and this excess post-oxygen uptake or utilization continues on for 48, sometimes 24 to 48 hours. This is your muscle biopsy. They come at you, if you think the needle for blood donation is big, you increase that some more, and that's the size of a biopsy needle. And the biopsy needle is in a sleeve that has a really sharp tip on it, and they ram that into the muscle, shove this other probe in, take a slice of the muscle out, and then you've got a little plug of muscle that you can then section and look at on the slide. So smaller muscles that are redder staining, those are going to be your slow twitch muscles. The ones that have a bigger cross-sectional area are going to be your 2B fibers, the fast twitch glycolytic fibers. The ones that are intermediate in reddish color and intermediate in cross-sectional area, those will be your type 2A fibers, fast twitch, oxidative, and glycolytic muscles. You look at when stimulation comes in, so action potential ceases and then muscle contraction begins. Fast twitch is producing peak muscle tension about 10 milliseconds after the stimulation, and then it's back to resting at about 30 milliseconds after stimulation. The slow twitch doesn't reach maximum tension development till about 50 milliseconds, and it continues to have muscle tension until it's about 100 milliseconds. So slow twitch generates power more slowly, and it also loses power or tension more slowly. The rate at which you can produce cross bridges which produce the muscle tension depends on the myosin ATPase speed. So that ATPase on myosin's head, the faster it can convert ADP to PI. So myosin ATPase activity, if it can convert ATP to ADP and PI fast, then it can form cross bridges fast. If it can form cross bridges fast, it can produce tension fast. So tension develops quickly, but then tension will drop off quickly. The myosin ATPase, if it's slow, then cross bridges will be formed more slowly because it takes longer for the ATPase to convert ATP to ADPI and get the high energy state for the next cross bridge. Slower the cycling of the cross bridges, longer time it takes to reach peak tension. Also takes longer for the sarcomeres to go back to resting level as well. So if you superimpose the 2A fibers in here, they probably peak out somewhere that take a little bit longer than a fast 2B fiber, but they'd be faster than a type 1 fiber. So here's a summary of everything about the muscles. So type 1, 2A, 2X or 2BX muscle fibers. So myosin ATPase activity, that means how fast does your ATP get converted to ADP and PI? Fast for 2A, but faster for 2X. And you look at the peer-reviewed journals, 2X fibers are faster than 2A fibers. They're both fast, but 2BX are fastest, and slowest are the type 1. Speed of contraction follows ATPase activity. The faster you can form cross bridges, the faster you can contract the muscle. So slow, fast, and fast. Resistance to fatigue. How fast do you run out of ATP? You don't run out of ATP at all in slow twitch fibers until they become ischemic, so it's pretty far down the road. Intermediate for 2A fibers because they don't have as many mitochondria as type 1, but they have more than type 2B, so their type 2B will be the most likely to fatigue, and these will be intermediate. Oxidative phosphorylation follows the mitochondrial concentration. So we have good numbers of mitochondria, can do a lot of oxidative phosphorylation. Type 1 is higher than this high type 2A, as it has more mitochondria. So here it would be the most, this would be many, and this would be few. Anaerobic glycolysis, that's going to be, these enzymes would be in the highest concentration where your glycolysis, you're only running glycolysis. Intermediate for 2A, you don't need that many glycolytic enzymes because you're not relying on glycolysis for ATP production because you've got these mitochondria. To store oxygen for oxidative phosphorylation, that will depend on the concentration of myoglobin in the muscle. It'll be highest in the red and less high, but still high in 2A, and lowest in the 2X fiber. You come back here, the reason this cell here is less red than that cell is because it has less myoglobin. So there is a difference in myoglobin concentration between type 1 and type 2A fibers. So it's high in 2A, but it's higher in type 1. This is reddest, this is less red, and this is closer to being white because very low myoglobin content. Glycogen content, if you're relying on glycolysis, you better store a lot of glycogen and have that glucose there. Intermediately, 2A fibers, they're not relying on glycolysis because they have mitochondria, so they don't have to have as much stored glycogen. And then the 2A fibers, least dependent on glycolysis, so the lowest content of glycogen. Yeah.
In Martini, their table like that, he kind of said that 2A was kind of more like 2B, and in that table it kind of says the opposite. You know, like it's obviously in the middle, but it was leaning towards 2B, and now it looks like.
For which one? For all of them?
For just in general, it's like it was like medium, this way, but in you know what I'm saying, in Martini it was in general it leaned towards the 2B, and in Sherwood now it's leaning towards 1. That's more similar.
It always is intermediate, so that's the best way to think about it. The type 2 fibers are glycolytic, but they're less glycolytic than 2A, but they're more glycolytic than 1. So It's a point of reference, I guess, which way you want to look at it. I guess the old timers would say, well, if it's a type 2 fiber, it's going to have a bigger cross-sectional area, therefore more power, and it's more like a 2B fiber than it is really a type 1 fiber. So you could describe it from that point of view as well. Just know that when it says high, that's oversimplifying. So when it says, oops, where is that? So when it says high, this is highest, this is high, but not as high. So there is this grade. It's not high. It's highest, then higher than 2B. Here, this is high, not as high, they'd say intermediate. So this is speed of contraction, this is fastest. This is fast, but not as fast as that. So there always is this intermediate type. There's always an intermediacy of the, I don't think that's even a word, but type 2A fibers are always not as oxidative as type 1, but more oxidative than B. So they're always intermediate. When you have either extreme being high, the 2A's are intermediate. Okay, so banding muscle contractions. I'm not sure what they call here.
The Z-line is a dense vertical line. The area between two Z-lines is one sarcomere. Z-lines are flat proteins that connect the thin filaments of two adjoining sarcomeres. The A band is a dark band. It consists of a stacked set of thick filaments along with the portions of the thin filaments that overlap on both ends of the thick filaments. The I band is a light band. It contains the portions of the thin filaments that do not overlap with the thick filaments. The H zone is a light area in the middle of the A band. It contains only thick filaments.
Okay, so contracting.
During contraction, the thin filaments on each side of the sarcomere slide inward toward the center. The contracted sarcomere is shorter than the relaxed sarcomere. The width of the A band remains unchanged because the thick filaments do not change length during contraction. The I band, which consists of the portions of thin filaments that do not overlap with the thick filaments, decreases in width as the overlap between thin and thick filaments increases. The H zone, the region in the center of the A band where the thin filaments do not reach, becomes smaller because the thin filaments approach each other as they slide inward. Notice that neither the thick nor the thin filaments decrease in length to shorten the sarcomere. Instead, the contraction is accomplished by the thin filaments sliding closer together between the thick filaments.
Okay, when they shortened the H zone, they actually got it down to the M line, which is a little misrepresented there. So H zone does narrow, but this gray in the middle, that's not the H zone, that's the M line. Okay, so motor units, number of fibers contracting depends on the number of alpha motor neurons that you stimulate. The muscle fibers per motor unit will vary, as mentioned earlier, anywhere from a few up to several thousand. The overall muscle fibers available to contract are dependent on the size of that individual muscle. So muscle like biceps, triceps, or pectineus, or plantaris versus gastroc, that kind of size difference. And then you have twitch summation, and you have the point at which you get maximum power output with those motor units recruited. So that will, you'll be able to summate twitches if the action potentials are being generated close enough together. So you have to have the stimulus frequency a high enough rate that you don't get calcium pumped back into the SR, or just minimal calcium pumped back into the SR. You've got to have some calcium remaining out in the cytoplasm in order to sum with the next stimulation. The length of the fiber at the beginning of the onset of the stimulation will determine how much tension can be developed. So that's your length tension curve. Where is length tension curve? Here. So the shorter your sarcomeres, less tension. The longer your sarcomeres, less tension. So optimum zone of overlap, you have the greatest force of contraction at that point. Yes?
Is there any benefit to the training those ends of the spectrum?
Oh, some people think yes, but at the extremes where you have less power being able to generate because there's fewer cross bridges, if your load is too high and you exceed the ability of the muscle to produce tension, that load gets transferred to your Golgi tendon organ, and those will actually turn off the muscle contraction as another attempt to protect you from damage. But if in that instant between the muscle force getting transmitted to the tendon and the Golgi tendon organ turning off the muscle contraction, you could already rip connective tissue. Connective tissue, if you don't know already, is very slow to heal. So talking 50% of connective tissue healed in 10 months, as opposed to, and that's just 50% repair. Muscle will heal, muscle cells will heal in a couple weeks, but the connective tissue damage, that would take months. And you try to go back to normal activity, you usually re-injure the connective tissue again. Okay, so extent of fatigue, the power muscles give out and less powerful muscles try to take over. You run the risk of damaging muscle and connective tissue. Type of muscle that's the most resistant to fatigue is type 1, but it also generates the least amount of power. Thickness of the fiber is the number of these contractile myofibrils in parallel. That will give you the greatest amount of strength. The speed is the sarcomere shortening in series, and that is the increase in speed is due to increase in the number of sarcomeres in series. If you have high testosterone, you tend to hypertrophy muscles more efficiently. Okay, so to Wednesday, we'll pick up with this complex.